In this video we will discuss about density and distribution of the proteins at the top of the hydrogel. Since they are developing a hydrogel, it's essential to know whether the proteins are distributed across the hydrogel or whether they are penetrated inside the cells. So for the studies that they have did, they use the anti-CD3 to be distributed across the top of the hydrogels, and then use T cells, T cell receptors, that is CD3 receptors on the T cells can interact with this anti-CD3 on the hydrogels. That can lead to the activations of the signaling pathways that is related to the CD3 activations. So this is what their idea. So before going into their experiments, they wanted to check whether the anti-CD3 are evenly expressed across the hydrogels, and also they wanted to check whether anti-CD3 will penetrate inside the hydrogel or not. So this is their experiments that they did it at different stiffness of the hydrogels, that is 2 kilopascal, 12 kilopascals, 50 kilopascals, and this is the control. That control does not have this anti-CD3, or it does not have streptavidin on the hydrogels. And so how they attach this proteins is that they took the hydrogels and first they coat the hydrogels with the streptavidin, modified streptavidin, and then they use biotinylated anti-CD3 to bind to these streptavidin that are coated on the hydrogels, and then they use PE anti-IG antibodies that can bind to this biotinylated anti-CD3 and express P fluorescence corresponds to PE channel. This is their plan to check whether CD3 is been distributed across the hydrogels or not. And when they did their confocal microscopy, they can see that the fluorescence intensity is same across the different stiffness of the hydrogels. And for the control where they do not use streptavidin, the fluorescence intensity is minimum. So this indicates that the interactions that they are expecting, that is anti-CD3, biotinylated anti-CD3 will interact with this PE anti-IgG, is seems to be correct, and their interactions are specific rather than the non-specific, because the control does not give fluorescence signals at a higher value. And then they check it for so in this panel A they checked whether the distributions are evenly expressed or not, and they check in the XY plane. And in the panel B they check the distributions in the Z plane, and the Z planes also they can see that only in the top layer of the hydrogels they are able to see the signals, but not in the bottom layer of the hydrogels. And for the controls they do not see any fluorescence signals. And the quantifications of this mean fluorescence intensity is shown in the panel C, where we can see that the fluorescence intensity does not vary too much with different stiffness of the hydrogels, but for the control the mean fluorescence intensity are lower. So their objective to study the T cells, the objective to activate the T cells on the hydrogel could be fulfilled by this approach, that is T cells are evenly spread out on the hydrogels and their density is also uniform. And from the previous figure we have understood that with the expressions or with the distributions of this anti-CD3, it does not impact significantly on the mechanical properties of the hydrogels. So this seems to be appropriate models in order to study their T cells interactions with the antigen presenting cells in terms of their mechanical properties or interactions due to the mechanical properties. So let me clarify this last statement. So their objective now seems to be this model seems to be appropriate to their objective, that is to understand the interactions between antigen presenting cells and T cells in terms of their mechanical forces, or antigen presenting cells interactions, mechanical changes that caused by antigen presenting cells when it is interacted with the T cells. So this is what their objective, and this system seems to be appropriate for their study. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.